Hello students. In this class I am going to discuss about the genes, genetic materials, nucleic acids and gene expression and protein synthesis. It will take 4 classes to complete the total syllabus of this topic, and in the first class I am going to discuss about gene, nucleic acid and nucleotides, and the second class I am going to discuss about the gene expression and protein synthesis. So let's start. The each cell of our body contains thousands of different proteins, and it is obvious to question that how these proteins are synthesized to which are the mainly the functional unit of life. And it was observed that each cell contains some specific structure that is known as nucleus, and in the nucleus the chromosomes are present, and the genetic information of heredity residing into these chromosomes, and they are chemically nucleic acid and histone proteins are there, which are containing the basic information of heredity. During 1940s, the DNA was discovered. It is one of the nucleic acid, and it upon discovery it was known that it contains the hereditary information, and several studies were demonstrated for the control of gene expression and DNA structure discovery. And now it is known to us that the DNA containing the genetic material which is transcribed into RNA, and the RNA, the proteins are synthesized, and this is known as the central dogma of life. So there are two types of nucleic acids. So one is the ribonucleic acid and another one is deoxyribonucleic acid, or DNA. Basically, the DNA and RNA are the polymers, but built from the monomers called nucleotides. And the nucleotides is composed of a nitrogenous base, a monosaccharide, and a phosphate group. So basically, the nucleotide is composed of the molecules carbon, nitrogen, hydrogen, phosphorus, and oxygen. These 5 molecules are involved in the making of the nucleotide molecules. So there are these nucleic acids are made up of nucleotides, and this is the nucleic acid structure. Here the phosphate group is joined with sugar moiety and the base, so nitrogenous base, and these nucleotide are polymerized to form the nucleic acid known as DNA or RNA. And DNA is present in the nucleus, and RNA is the cytoplasmic nucleic acid. Their function is the storage of genetic material and transmission of genetic material, and they also control the protein synthesis of cell. So this is the sugar molecule, and the sugar is basically a pentose sugar, and the two prime carbon containing the hydroxyl group gives rise to the ribose sugar, whether the 2 prime goes carbon source 2 prime carbon devoid of the hydroxyl group gives rise to the deoxyribose sugar, and the presence of these type of sugars respectively gives the name RNA and DNA. There are 4 nitrogenous bases in the nucleic acid. These are classified into 2 groups. One is the purine, another one is the pyrimidine. And the purines are A and adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. And the DNA contains A, G, C, and T, and the RNA contains A, G, C, and U. So these are the structures of pyrimidines and purines. The nucleotide is formed with the joining of adenine, with the joining of nitrogenous base with the pentose sugar and the phosphate group, and during this type of joining the covalent bond is formed in between the moieties, and during this type of reaction the water molecule is evolved, and thus the ester bond is formed in between the nucleotide moieties, so that the growing nucleotide chain can form the nucleic acid polymer. So this is the structure of DNA and RNA. This is the basic structure, which is containing the ribose sugar, nitrogenous base, and phosphate group. And the sequence of these nucleotides is known as the primary structure of nucleic acid. Because why this is called primary structure? Primary structure means the sequence of nucleotides. And in case of DNA, it is double stranded of these nucleotides chain, and in case of RNA, this is single stranded. So this is the structure of DNA and RNA, which is common in both of them. And basically the structure is a linear polymer of nucleotides. So this is the linear polymer of the nucleotides, which is shown in this picture. Now DNA can also ordered in an arranged in the ordered structures to form the secondary structure, which is known as the double helix model. And this structure was first proposed by the J.D. Watson and Francis Crick, and for this discovery they were honored with the Nobel Prize. And this type of double helix is a secondary structure of DNA in which the two polynucleotide strands are coiled around each other in a screw-like fashion. So this is the figure where Crick and Watson describes the structure of double helix nature of DNA. And this is the DNA double helix structure, which is basically a basically an anti-parallel structure. If the red strand is going in one direction, the blue strand is going to the another direction or the opposite direction. So it is known as the anti-parallel structure of DNA. And this type of structure gives rise to the stability of the DNA double helix by the formation of hydrogen bonds in between the base pairs of particular adenine and thymine and cytosine and guanine. And we can found that that the hydrogen bond is formed between adenine and thymine, and also for the hydrogen bonds are formed in between guanine and cytosine. And we also found that the adenine and thymine can be base paired with 2 hydrogen bonds, and that's for cytosine and guanine there are 3 hydrogen bonds. So the higher structure of DNA also can be found. The DNA double strand can be coiled around the protein known as the histone protein for the compact structure generation of chromosome. And histones are rich in the basic amino acid lysine and arginine, those whose side chain are have a positively charged containing positively charged amino acid. And as the DNA is negatively charged, they will form the interaction, electrostatic interaction with these positively charged histone proteins, and they form the unit known as nucleosome. Nucleosome is a core of 8 histone molecules around which the DNA helix is wrapped. The nucleosome are further condensed into the chromatin structure. Chromatin fibers are then organized into loops, and the loops into the bands provide the structure of chromosome. So let's see how this is formed. So this is the double helix structure of DNA, and it upon coiling on the histone protein, it will form the nucleosome. The nucleosomes are arranged on as the bead on a string to form the chromatin molecule, and the chromatin molecules are further organized into the formation of the solenoid structure. When the solenoids are organized in such a way to form the mini band, and this is the mini band structure is basically the cross section of a chromosome, which is depicted here, which contains basically the loops and coils of DNA double helix wrapping around the histone proteins. So basically this is the chromosome, which is which is made up of the super solenoid structure, and the solenoid structure is made up of the nucleosome, and the nucleosome is made up of the histone protein and DNA around the histones, and this is the structure of DNA double helix. To basically this structure is formed to accommodate the large amount of genetic material DNA in the nuclei of the cell, and that is basically possible by the coiling around the proteins, histone proteins. And this is the micrograph of a chromosome. This is the basically metaphase chromosome, which can be seen under light microscope with some stain. And during the cell division, the chromosomes are condensed so that upon staining, it can be visualized under microscope. So there are three differences between the structure of DNA and RNA. The DNA bases are A, G, C, and T, and the RNA bases are A, G, C, and U. And the sugar in DNA is deoxyribose, and in the case of RNA, it is ribose sugar. The DNA is always double-stranded, and there are several kinds of RNA, which are most of them are single-stranded in nature. So let's see how many types of RNA are there. There are several classes of RNAs, as well as I forgot to tell you, there are several classes of DNA also. Basically, we can found in nature there are three types of DNA known as aDNA, bDNA, and zDNA. So these are the there are some RNA molecules present in our nature. Those are transfer RNA, ribosomal RNA, messenger RNA, small nuclear RNA, microRNA, and small interfering RNA. The functions of these RNA are given below. Basically, the transfer RNA transport amino acid into for the protein synthesis. The ribosomal RNA combines with proteins to form ribosome. This is the site for protein synthesis. The messenger RNA directs the amino acid sequence for the protein synthesis. The small nuclear RNA basically processes the initial mRNA to its mature form in eukaryotes. The microRNA affects the microRNA and small interfering RNAs. They both affect the gene expression. Then this is important for the growth and development. So this is the structure of mRNA or messenger RNA. This is the primary structure of mRNA, and this is the secondary structure of mRNA, where the if there is a complementary region can be found, due to the complementary nature, the hydrogen bond can be formed in between them. So the messenger RNA can be super heliced in between itself. And this is the structure of tRNA or transfer RNA. This is the basic clover leaf model of structure, and you can see that the hydrogen bond is present when the in between the stems, where the complementary regions are there, and where the complementary region is not found, the structure is that region is ultimately conformed into the loop-like structure. And this is the 3D structure of tRNA. This is the structure of ribosomal RNA, and it is the most abundant RNA in the cell. So and you can find that there are several stem and loop structure, and this structure is too much complicated, and this is the most complicated structure of RNA molecules. So students, in this class I have completed the first portion of the module, and I think it will be helpful for your exam purpose. Please go through the slides, and if you have any question, you can ask me later. Thank you.